Amino acid. Amino acid, the acid group is always C double bond O, OH. So there is one carbon. To that carbon, there is an acid group attached. Acid group is always C double bond O, OH. And amino group, let me make it more clear. And amino group is always NH2 group, and there is a hydrogen attached to this carbon, and then to this carbon is attached an R group. R is just a symbol, you can say, which means anything can be attached here. For example, methyl, ethyl, benzyl, anything. Different amino acids will have different R group. So what an amino acid is? Any compound which has got amino group and acid group attached to one carbon. That is called amino acid. It is amino also, it is acid also, provided the amino group and acid group, they are attached to the same carbon. This is the carbon to which it is attached. On one end it has got amino group, on other end it has got acid group. Carbon has 4 valencies always, so one of those will be hydrogen and then an R group. So for an amino acid, this must be common in all amino acids. They must have amino group, acid group and hydrogen. What different amino acids differ in is their R group. Different amino acids will have different R group. Okay, and it is a concept of chemistry that any carbon which has got all 4 different groups attached to it, acid group, amino group and R group and H group, all are different. All different things are attached to it. Any carbon which has got all different things attached to it, that is called chiral carbon. Carbon which has all different things attached to it. That will be called a chiral carbon. Usually on a chiral carbon, we use a symbol like this. So a star. So this is a chiral carbon. So presence of chiral carbon, presence of amino and acid group on the same carbon, these are the features of an amino acid. Is the structure of amino acid clear? Okay. Next is classification of amino acids. Classification of amino acids. Amino acids are classified according to various parameters. I mean, in different ways they are classified. One way of classification is they're classified into essential and non-essential amino acids. Essential and non-essential amino acids. What do we mean by essential? They are essential in diet. just like we talked about in fatty acids. They're essential in diet. Why they're essential in diet? Body cannot synthesize them, so they must be obtained from diet. And non-essential amino acids are may not be present in diet, may not be present in diet, body can synthesize them. That is the basic difference between essential and non-essential. Now you must be knowing that there are 20 amino acids in total. In the whole biological system, may it be humans or animals or plants, there are 20 in total. And they arrange and align themselves in different sequences to form so many different types of proteins. So all the proteins, they just differ in different arrangement of these amino acids, nothing else. We were talking about the classification of essential and non-essential. Firstly, let's talk about essential. Let's write essential on this side. For essential, you have one mnemonic given in all your books, that is PVT, Tim, Hall. P stands for phenylalanine. V stands for valine. T stands for threonine. Another T stands for tryptophan. I stands for isoleucine. M stands for methionine. H stands for histidine. A stands for arginine. L stands for lysine. And another L stands for leucine. So we have phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, histidine, arginine, lysine, leucine. All others are non-essential. So you need not memorize the names of non-essential ones. Just memorize essential, and if you see anything other than essential, that of course will be non-essential. And these are 10 in total if you see. Out of these, histidine and arginine, they're called semi-essential. They're not completely essential, but semi-essential. What do we mean by semi-essential? Semi-essential means they become essential at some stage in life. So usually they are non-essential. That means the body can synthesize them, but at some particular stages they will become essential. That means, let's say body is synthesizing 5 and the body requires 20. So even if body is synthesizing 5 is not enough, so 15 must be obtained from diet. So that way they become essential, even though body can synthesize them, but still they should be obtained from diet, at least at some stages of life. That is why they're called semi-essential. They can be synthesized, but also essential. What are those stages? Stages can be rapid growth, pregnancy, etc. So this is one type of classification. Then amino acids can also be classified into acidic. Another classification is they can be classified into acidic, basic, and neutral. Acidic are also called negatively charged. Basic things are also positively charged, so they can also be written as positively charged. Neutral things will be neutral for charge also. So acidic, basic, and neutral. Acidic amino acids are glutamic acid, which can also be called glutamate, or aspartic acid, which can be also called aspartate. So they ask you which of these is an acidic amino acid. This is an exam question. But if they write glutamic acid, it will become very obvious that this is acidic. The name itself has acid in it. Similarly, aspartic acid. So in order to make it less obvious, they write glutamate instead of glutamic acid, or aspartate instead of aspartic acid. Positively charged or basic ones or alkaline ones are histidine, arginine, these are the two semi-essential ones, and lysine. Histidine, arginine, lysine, they are basic ones or the alkaline or the positively charged. And rest of everything will be neutral, neutral by charge as well as neutral by pH. Is that clear? Then there is another thing they ask, which of these is sulfur containing? Sulfur containing ones are cysteine, cysteine, and methionine. Cysteine, cysteine, methionine. So everything else will be non-sulfur containing. Then they can ask you which of these is aromatic. Aromatic is those which have a closed ring structure. Examples of aromatic are tyrosine, tryptophan, and phenylalanine. So these are all exam points. They ask you these examples. Then there are two amino acids which are non-protein forming. We have 20 amino acids which take part in protein formation, but there are two amino acids which are non-protein forming. That means they don't take part in protein formation. Then why are they called amino acids? Because they have the same structure. They have amino group, they have the acid group attached to the carbon, and hydrogen is there, and R group is there. So chemically their structure is same as that of amino acid, the amino group, acid group, but they never take part in protein formation. Those are ornithine and citrulline. And these take part in urea cycle. They are part of urea cycle. They never take part in protein formation. They only take part in formation of urea. So they're part of urea cycle. So these are different types of amino acids. I know it's a lot of memorization, but the essential, non-essential you must know. Acidic, basic you must know. Sulfur containing you must know. And the non-protein forming, these you must know. Only thing you can skip is the aromatic one. I've rarely seen any question about aromatic, but all rest of the four you must know. They do ask you. Then there are some amino acids which undergo hydroxylation. Amino acids which can undergo hydroxylation, this is again very important exam question, are proline, it can become hydroxyproline, then glycine, it can become hydroxyglycine. And even lysine, that can become hydroxylysine. So that's all about the different types of amino acids, their classification. As I told you that acid group is negatively charged, basic group is positively charged. Amino acid has got both acid and basic group. So they have positive charge at one end and negative charge at the other end. But depending upon what kind of R group is there, in some amino acids, positive charge is dominant. In other amino acids, negative charge can be dominant. But both contain, all the amino acids contain both types of charges. Depending upon what kind of R group is there, either the positive will become stronger or negative will become stronger, or both will remain equal. Next, we have to do the breakdown of amino acids or degradation of amino acids. Now, amino acids have got an amino group. breakdown of amino acids. Amino acids, they have an amino group. When this amino group is removed, it becomes ammonia. Ammonia is very toxic. So it should be immediately converted into urea somehow, which will be excreted in urine, which will be passed out in urine. So out of the whole amino acid, firstly we'll talk about how the amino group that becomes ammonia and that is converted into urea and urine. The rest of the acid part which is left behind we'll talk about later, because the management of amino group in itself is a big cycle. Amino acids undergo transamination. Transamination means transfer of amino group from one amino acid to the other. How that happens, let me show you the picture. Do you see the screen again? Okay, now listen to this carefully. Please stop writing for some time. I'll put this picture on document. In fact, it is already there on your document. As the word itself says, transamination, that simply means transfer of the amino group from one thing to the other. Firstly, why transamination is happening? Why not amino group just being removed and converted into urea? Why it has to be transferred? Because urea cycle occur only in liver. So only in liver the amino group can be removed and converted into urea. For all other tissues, it must be transferred to something. Then it should be transported to the liver, and then only liver will make urea. So for all other tissues, if amino group is left alone, ammonia will form and that will be very toxic. So in all the cells of body, only transamination occur, that is transfer of amino group from one amino acid to the other, or one amino acid to some other compound. Only in liver it can be removed and converted into urea. So you can write in your notes, in all tissues, transfer of amino group take place from amino acid to some other compound. Only in liver the amino group can be removed and converted into urea. Then please see this clearly, how this actually happens. This is an amino acid. Any amino acid, they have generalized formula written. You have the acid group, you have the amino group. I told you acid group is negatively charged because it can lose its H positive and become negatively charged. Whereas amino group, NH2 can gain an H positive and it can become NH3 positive. So it is just the same amino acid shown in ionized form, nothing different. We can also write it as C double bond OH, and here we can write as NH2. Okay, it's just that this H positive from here jump here. So we are left with, after the jump, we are left with negative charge here and positive charge here. So it is the same amino acid shown, acid group, amino group, and hydrogen attached to one carbon, and then is a R group. Now this amino acid cannot get rid of the amino group as such. It can only transfer it to something else. Something else which helps in this transfer is alpha ketoglutarate. Remember, alpha ketoglutarate was one intermediate of Krebs cycle. So every cell which has got mitochondria has Krebs cycle running in it. So it will have alpha ketoglutarate. This alpha ketoglutarate is a keto acid. What do I mean by keto acid? C double bond O group is called keto group. So this is the acid group. Next to acid group is a keto. C double bond O present next to the acid group. That is why it is called keto acid. So alpha ketoglutarate is a keto acid. What happens is these two compounds, they exchange the amino and the keto group. This is the keto group. The amino group is already highlighted. So they both exchange such that amino group comes here and keto group goes here. This is the second carbon. It was initially having amino group. You can see R and then the second carbon and then the acid group. It was having amino. Now the same R and the acid, but the second carbon has double bond O. It has become keto. Keto acid. From amino acid, it has become keto acid. Whereas this was a keto acid initially, it gained amino from this. And now, first is the acid group, to the second carbon, it was a keto, now it has an amino. Rest of it is same. So what has happened here is, again, amino group went here and keto group went here. So they have exchanged their groups. The amino group has got transferred from amino acid to alpha ketoglutarate. That is why it is called transamination. Do you see what has happened up till now? Is that clear? Yeah. So alpha ketoglutarate, after gaining the amino group, will become amino acid, right? Now it is amino group next to acid. So it will be called amino acid, which is glutamate or glutamic acid. You know, its other name is glutamic acid. This is one of the acidic amino acids, glutamic acid or glutamate. This is what is called transamination. Next, what happens is, I'm going to remove this part for now because this part happens in liver, so I'll talk about it later. So next what happens here is, Now this glutamate also want to get rid of the amino group. It cannot leave amino group as such. Ammonia will form, that is toxic. So what happens is, every cell has got pyruvate. It is the product of glycolysis. Pyruvate is a keto acid. We didn't study its structure because structure of all intermediates is not needed. That will be too much. But just for your knowledge, it is a keto acid otherwise. The glutamate will give amino group to the pyruvate and convert it into alanine, which is an amino acid. And itself, it will regain the keto from pyruvate and become alpha ketoglutarate. So another transamination has happened. Pyruvate gave its keto, so keto came here, and glutamate gave its amino, so amino came here. Pyruvate becomes an amino acid, whose name is alanine, and glutamate becomes alpha ketoglutarate again. Now, if you remember the fates of pyruvate, if you remember different fates of pyruvate we did, I told you most common fate is acetyl-CoA, but it can also get converted into lactate, oxaloacetate, and alanine was one of the fates. Remember? This is how pyruvate can become alanine. If it undergoes transamination, if it gains amino group, it will become alanine. Is that clear? So any amino acid is giving its amino group first to alpha ketoglutarate, and then this alpha ketoglutarate becomes glutamate. This glutamate gives its amino group to pyruvate, making it alanine. Now, this alanine has the capacity to diffuse out of the cell. It goes out of the cell into blood, and along with blood, it will reach liver. Glutamate cannot go out directly, so glutamate will not be helpful. So glutamate has to further give the amino group to pyruvate, making it alanine. Alanine is capable of diffusing out of the cell, so it will diffuse out of cell and come into the blood, and along with blood it will reach liver. You can write down this if you want. You can make your notes, whatever. I hope it is clear up till here. Do I move on? Okay. Now, once this alanine is in liver, this is an amino acid, right? It will further undergo transamination and give the amino group to alpha ketoglutarate again. Alpha ketoglutarate has got keto group, which will be given to alanine, making it pyruvate again. So alanine will become pyruvate after gaining the keto group again. And the alpha ketoglutarate will become glutamate, the amino acid. but now it need not give it to something else. Now it can directly leave its amino group because now it is inside liver. So this is transamination. Here the transfer of amino group happened and it has got exchanged with keto. So this is transamination. But the glutamate, now this part of reaction will come up, this which I left. I'll write it this side only. So now the glutamate becomes alpha ketoglutarate. With help of glutamate dehydrogenase, NADH is undergoing any change, that is not important. And it is getting rid of the amino. Now this is not giving the amino group to something. This is not gaining the keto group from something. It is just leaving the amino group and becoming alpha ketoglutarate. This step is not transamination. It is deamination. Deamination. The amino group is just getting removed. And why is it possible? Because it is in liver. Why this process is possible? Because it is happening in liver. In liver, it is possible. Why? Because this amino group, NH4 is just dissolved form of ammonia only. NH3, when it dissolves in water, it will present as NH4 positive. So this is ammonia. In other cells, ammonia cannot be left because it is very toxic. But in liver, it can be left because it will be immediately entrapped into urea cycle and it will be converted into urea, which will be passed out of body in urine. So deamination in liver is possible. So glutamate will just leave its amino group and become alpha ketoglutarate. Whereas in other cells, it was not possible. Whereas this pyruvate, it will become acetyl-CoA and go into Krebs cycle, or it can do whatever it wants. This is how the amino acids get rid of the amino group. First, in every cell, wherever the protein breakdown is taking place, they will just transfer the amino acid to alpha ketoglutarate, making it glutamate. Glutamate further transfers it to pyruvate, making it alanine. Alanine will come out in blood and reach liver. In liver, the alanine again give it to alpha ketoglutarate. Just like in every cell, any amino acid can give to the alpha ketoglutarate. Similarly, here also any amino acid can give to the alpha ketoglutarate. So same thing is repeating. Once alpha ketoglutarate gain it, it becomes glutamate, but now it need not give it to something else. It will just undergo deamination, leave the amino group alone, which will be picked up by urea cycle. We'll do the urea cycle in detail and then convert it into urea, and then that is passed out in urine. Is that clear? What happens to the amino group? Okay, I'll put the previous picture is already there on your document. I'll put this one also. I hope you see the screen. So you see the previous picture that the alanine go into blood and liver, and then in liver what happens? This alanine undergo transamination and then deamination, and urea cycle will happen. After removal of the amino group, when the amino group is gone from amino acid, we are still left behind with keto acid. So it is not complete breakdown of amino acid yet. We are just handling one group of amino acid. Rest of the amino acid is still there, and we will have to study what happens to the rest of it. But before that, let's see how this ammonia which is released become urea. So for that we have to study this urea cycle. Urea cycle, remember, it occurs only in liver. One thing. Second, it occurs partly in mitochondria and partly in cytoplasm. This is very important. Till now we have been studying that either a reaction happens in cytoplasm or in mitochondria. But this is the first one which is partly happening in mitochondria and partly in cytoplasm. So these two points are very, very important. Firstly, urea cycle happens only and only in liver. And secondly, it occurs partly in mitochondria and partly in cytoplasm. What happens is, this is the ammonia which was released from deamination. It combines with bicarbonate ion. This is inside mitochondria. Inside mitochondria, the ammonia combines with bicarbonate in presence of carbamoyl phosphate synthase to form carbamoyl phosphate. So carbamoyl phosphate is formed. This carbamoyl phosphate, it combines with ornithine to form citrulline. Remember these ornithine and citrulline, I told you non-protein forming amino acids, and they occur only in urea cycle. So this is their role in urea cycle. They are intermediates of urea cycle. So this carbamoyl phosphate which is formed combines with ornithine and becomes citrulline. Citrulline comes out of mitochondria in cytoplasm. This is cytoplasm, where it is becoming arginosuccinate. Arginosuccinate splits into arginine and fumarate. So you can see it is splitting into arginine and fumarate. Arginine further acted upon by arginase, it splits into ornithine and urea. So this urea will be passed out it will be filtered from blood by kidney and passed out in urine. So again, you need not know the cycle, you need not know all the intermediates. Points which you have to know is it occurs in liver. Second point, it occurs in partly in cytoplasm and partly in mitochondria. Thirdly, two non-protein forming amino acids are intermediates Their names are ornithine and citrulline. And lastly, which is quite frequent exam question, they ask you from which intermediate urea is finally formed. Urea is finally formed from arginine. Arginine finally split into urea and ornithine. So from what urea is formed? Urea is formed from arginine. You know arginine is an amino acid. Remember histidine, arginine. We have been doing that. Then another exam question they ask you is which of these amino acids is not part of urea cycle? And they give you options. Option A, ornithine. Option B, citrulline. Option C, histidine. Option D, arginine. So of course ornithine, citrulline and arginine are parts of urea cycle. Histidine is not. So you must have the gross idea of the cycle, where it is happening, how it is happening and what it is doing. But you need not know the enzymes and all the intermediates and reactions, etc. Urea in the end comes from arginine, that you must know. Then which all amino acids are seen in this cycle, that you must know. And where it occurs. That's it. So you see aspartate also here. You know it is aspartic acid. It is also an amino acid. Even though it is not intermediate of the reactions, it is formed in the process. It is being formed. So they might ask you which of these has no role in urea cycle, which of these has. So in options, aspartate also might be there. Just keep in mind that this also is there, but it is released. It is not any intermediate or product. Is that clear? I think we stop here for now. Next, we have to do the synthesis of amino acids, which is again a whole big topic. So I'll begin it next class. That's all for now. Lots of memorization in today's class, but you do get so many questions out of those. So make sure you not only understand, but also memorize all those points. That's all for now. See you guys tomorrow. Bye.